of the lecture series for Bio 40A. I am going to start the chapter which is entitled The Cellular Level of Organization. I believe this is chapter 3. And as we take a look at the objective set questions, you will see a whole list of areas that I'm going to lecture on. So I think what's going to happen today in today's lecture is I'm going to go through and cover these questions that you see listed here. They continue on into the next page. If I can get this done. Okay. And I'm going to stop right here with define the term organelle. So all of this material will be on a second presentation, on a second lecture presentation. So we're going to cover that first part, which is, as you can see, most of the questions, and then we'll go through and look at the cellular organelles, describe them, their function, and that will cover the chapter. So you'll notice, if you're looking at the chapter, that there is a lot in the chapter that we're not going to cover. So mitosis, meiosis, cell cycle, all of these things are not going to be covered and won't be on the exam. So just those things in the objective set question. All right. Well, let's begin. And let's talk about a cell. So let me bring this up. And this is one of the first pictures that are presented to you in this chapter. And this is let me do it this way. There you go. So this is, your book refers to this as a typical cell. What does that mean? Well, what it's saying is that this cell represents all the possible permutations and structures that you might find in any one particular cell. So you would not find a cell that looked like this in your body. So what they've done is made a composite cell, and they've shown you all the various structures that we're going to end up talking about. So just to give you a sense, we'll be talking about microvilli, we'll look at cilia, and we'll look at all of these structures that are defined as organelles, such as the nucleus, the endoplasmic reticulum, Golgi complex, mitochondria, and vesicles. So a variety of things we'll look at this. So this is a typical cell. It doesn't represent a particular cell in the body, but it represents a composite of all the possibilities that there are in your body. So we'll refer to this later on. Come back to it. Okay, so let's begin then our lecture, and we will start off with this title, Introduction to the Cell. Okay. So when we look at cells and collectively and analyze them, what we find is that there are 4 basic principal parts. So the cells are composed of 4 principal parts. And I'll go ahead and list them, and then we'll look at the details of each one. So the principal parts are, number 1, the cell membrane. Number 2, something that is referred to as the cytosol. Number 3, organelles. And the 4th one, as you will see, is the nucleus. Now, technically, the nucleus is an organelle, as you'll see your textbook refers to it as such. But it is such a special organelle and has a special function, such a special function in the cell, we pull it out and look at it as a major part of the cell. So cell membrane, cytosol, organelles, and nucleus. Those are the four principal parts. Let's describe a little bit about each of those areas, and we'll begin with the cell membrane. We're going to talk a lot more about the cell membrane a little bit later on in the lecture, so we're just drawing a quick picture here, and then we'll go into the details later on. So this is really the plasma membrane. That is the technical term. That is the proper way to refer to it. But so many people simply simplify that to cell membrane. I'll probably end up doing that too throughout the lecture. But keep in mind, the proper way to refer to this is the plasma membrane of the cell. Okay? And the function of this is many-fold, but the general function is to create a space within which the cell can control. So basically, this is going to divide the outside world from the inside world of the cell. It's the border between the outside and the inside. And of course, the outside in your body is what? Extracellular fluids, as well as all that is found in those fluids. And the inside is the intracellular fluid, as well as organelles and other structures that we find. So this is the border between the outside world and the inside world, and it has a very complex structure, which we'll talk about, and it really allows the cell to control what happens within the boundaries or the border of the plasma membrane. All right, so that's the membrane. That's one of the four principal parts. The second part that we listed was the cytosol. So the cytosol is a thick, semi-fluid intracellular fluid. Now that's a little bit redundant, but it's a thick semi-fluid within the cell. You can think of this, the consistency of this, as like a thick minestrone soup, and it contains a lot of things. It contains, or dissolved within it, you're going to find proteins and nutrients, gases such as O2 and carbon dioxide, ions, and a host of other things are dissolved within that, plus the organelles. Okay. Well, actually, technically, let me make that more clear. So there's another term which I will define, cytoplasm. And the cytoplasm includes the cytosol, which we've just described, plus the organelles. So I was about to make a mistake and lump the organelles into the cytosol, but they're actually separate. And when we combine that together with the cytosol, what we're really talking about is cytoplasm. So the terminology is quite similar between cytoplasm and cytosol, but this is the fluid part. And these are plus the organelles. And that those two things together, the fluid part and the organelles, are referred to as the cytoplasm. Okay. Let's move on and talk about the organelles. And here we're going to spend a whole lecture talking about these, so I'm not going to go into a lot of details at this point. So the organelles are highly organized subcellular structures with usually a characteristic shape and that perform specialized functions within the cell. Highly organized subcellular structures, usually with a characteristic shape, that perform specialized functions within the cell. And as I said, we're going to spend a whole lecture describing exactly what all of that means. And that's where we're going to let it rest for right now. And the fourth and final part, major part, is the nucleus. And hopefully most of us have some sense of what this is. So this is a structure that contains the DNA, or I should say most of the DNA. I cannot say all of the DNA because there is, as we'll see, structures inside the cell that also have DNA, other organelles, but most, by far, most of the DNA is contained in the nucleus. This is where we find our genes that code for different proteins. And this is also the site of ribosome synthesis. So we build here in this structure ribosomes, which we'll talk about their function inside the cell later on. Okay, so that's the nucleus. Well, let's go on now and start focusing a little bit more in depth with many of these things that we've just listed as major parts of the cell. Okay. And the first thing that I would like to do is take a look at the plasma membrane. And to do that, got a nice little overhead. And we'll spend some time looking at this, and then we'll get some more notes, and then we'll come back and look at this to emphasize what we wrote in our notes. So this is a plasma membrane of the cell, and as you can see, it's fairly complex. There's a lot going on here. And just to kind of quickly identify the major parts that we'll end up talking about. So hopefully you recognize these molecules here. There is a double layer, and these are the phospholipids. So we lectured on that when we did the chapter on chemistry. Excuse me. So here is my phosphohead, and there is my two fatty acids. And one of the things I want to point out to you is this there, right there. You see, that's not a straight line. It has a little bend in it. And that's because we have a double bond right there. And if you recall, we talked about that. Fatty acids, when they have these double bonds between adjacent carbons, that causes the chain to stop being straight and puts a little angle. And that's going to be important. We'll talk about that importance later on. We also see embedded within those fatty acids another lipid. This is cholesterol. We see that embedded within the membrane, and we'll talk about the significance of that later on. So this is the lipid bilayer, and keep in mind that this layer is produced automatically. If I take a jar, a beaker full of these phospholipids, and I just pour it into water over time and fairly quickly, those phospholipids would organize themselves in this fashion that you see right here with the lipid bilayer, because this phosphohead wants to mix with water. It's hydrophilic, and the tails are hydrophobic, and so they sequester themselves. So you have water facing here, and water being excluded in this region here where the fatty acids are. We'll just go along the line and identify some other things, which we'll come back to. Here we have a glycolipid. So what is a glycolipid? Well, that's a lipid upon which we attach a sugar. And we see those spread out through the cell and poking out. We'll talk about what that all means later on. We have here what is called a peripheral protein. And that is a protein which is only on one side of the membrane. So in this case, it's on the outside. And here we have another one, a peripheral protein. This is on the inside. You notice it doesn't go across the membrane. It's only found on one side or the other. We'll define that, talk about peripheral proteins in a little while. And then we have a whole series of proteins which cross from one side to the other of that membrane. And some of them have sugars attached, making them glycoproteins. We'll talk about the significance of that. Here we have an ion channel. In this case, it's a gated ion channel. We're going to discuss that a little bit later on. And then here's another protein. So these three proteins, which span the whole distance between one side and the other side of the plasma membrane, are referred to as integral proteins, or transmembrane proteins is another way to talk about it. So there again is my lipid bilayer. Okay, so we'll come back and look at this picture as we go through and discuss the membrane. So let's get that started. So we're going to look at the plasma membrane. Of the cell. As I mentioned, this is going to be the gatekeeper of the cell. It is going to control what goes in and what goes out of the cell. Okay? And so the way we describe the structure of the plasma membrane, we've come up with a model to describe the way it's put together and its behavior. And this is called a fluid mosaic model. Fluid mosaic model. One way to visualize, and I'll get into the details in a moment, but one way to visualize what that means, the fluid mosaic model, is if you can think of the ocean with icebergs floating in it. Now, the surface of the ocean is not static. You've got these waves, you've got the undulating surface of the ocean, and then sitting in the ocean, spaced out different distances from each other, are these icebergs, and they are floating in that dynamic moving surface that we call the ocean. So to apply that example to what we're talking about here, the ocean would be the plasma membrane. So let's come back and look at that quickly. And let's do that in English. Right side up. And in English. There we go. We got it. So in this case, this would be the ocean. And here's my icebergs. Okay. This is undulating. It has flexibility. It's stable and it has flexibility. And we're going to talk about how that property comes to be in just a little while. But this is the idea that is captured in this term, fluid mosaic model. Fluid because it has flexibility, there's movement in there. Not only the membrane is flexible, but the proteins can move around. Unless they're anchored in place by some protein, they can act like icebergs and literally move around into different locations. So mosaic because we have these proteins that are spread out in a mosaic-like fashion within the membrane. Okay. So there is your basic model. Let's talk about the membrane lipids. because they are, of course, extremely important in giving us this this model, this fluidity in the model. So the fluid term comes from the behavior of membrane lipids. So when we look at the lipids, we have three major types of lipids, and we'll describe each one. These are the major ones, and we have smaller amounts of others, but let's talk about, I'll list them and then we'll talk about them in detail. So the first one is phospholipids, and these we have described in the previous lecture on cells, phospholipids. Second type of lipid is cholesterol. And a third type is glycolipids. So those are the three major types. And just to give you an idea of their relative amounts, their relative abundance, phospholipids represent around 75% of all the lipids that make up a membrane. The cholesterol is around 15 to 20%, probably closer to 20%. And the glycolipids represent around 5%, so a smaller percentage. Okay. So those are the major lipids. Let's talk about each one and talk a little bit about how they behave. Okay, so first the phospholipids. And as you recall, just quickly reviewing, we have this easily made representation of a phospholipid. So we have our polar head, and then we have our non-polar tails. And this is composed of a phospho group, so it has a charge, and the nonpolar tails are fatty acids. Yeah. And what we're going to do is we're going to arrange them in the phospholipid bilayer. We're going to produce what is called a phospholipid bilayer. And of course, bi means two. So let's go ahead and draw ourselves a quick little picture to represent that phospholipid bilayer. And we're going to do this. Here is going to do a bunch of heads here. I'll line those babies up here. And then we're going to have correspondingly on the other side there, and then we're going to bring in some tails. Okay, so let's do that. And just to make them different, we'll see this. And then I'm going to put, now remember, when we have these fatty acids with a double bond, we get that little bend, and that is going to be significant, as we'll see. Okay, so we have this kind of arrangement, and let's go ahead and pull out one of those okay, and what we have here, that point right there is a double bond. Between two carbons. In other words, we get this arrangement, whereas the rest of the chain is constructed like that. But when we put that double bond in, we get this bend. Instead of getting a straight chain for the fatty acid, we get this little bend, and that's what I've represented here, spaced out there. Okay. Now, let's come back to that image and just reinforce that idea by looking at the image again. And so here we are. And I'm going to zoom in a little bit now. So there is the lipid bilayer. Lipid bilayer. And they've shortened it. It's phospholipid bilayer, but that's okay. Lipid bilayer is fine. And you can see spaced out these little dog legs for some of the phospholipids. Okay. All right. So here I am emphasizing this. What is the importance of those things? And the answer to that is that membrane fluidity. Remember, we have this fluid mosaic model. Is produced by two factors. Number one, the double bonds within fatty acids. And what that does is, let me give you two examples. Let me give you two fatty acids right here, and neither of which have a double bond. Those two fatty acids can get quite close together because there's nothing pushing them apart. But if I have a fatty acid with the bonds, see, they get pushed further apart. So this becomes stiff, and this is less stiff. And so some of the fluidity in the membrane is the result of these double bonds and these little kinks in the fatty acids pushing the molecules further apart from each other and making them more fluid and more flexible. Okay. The other factor that's going to add to the fluidity of the membrane is cholesterol. And cholesterol tends to stiffen the membrane. So what we're trying to do is strike a balance between flexibility and strength and holding the whole membrane together into a coherent unit. And over time, through evolutionary processes, that balance has been struck very beautifully. And it's between the double bonds in the fatty acids and the cholesterol, we get just the right amount of flexibility and the right amount of stability of that membrane. Okay? So we've got the phospholipids, and what their purpose is to actually create the lipid bilayer, as we talked about. Okay. What about the glycoproteins and cholesterol? What is their job? So let's talk about cholesterol. We already mentioned it. We're simply going to basically repeat this. So cholesterol is a ring lipid. So remember, it's composed of rings. And what it does is it strengthens the membrane. The glycoproteins, glycolipids, I'm sorry. The glycolipids are signal molecules. And what they do is they're only found on the exterior. And they are kind of marker lipids. And what they do is they allow your cells to identify themselves to such things as the immune system and so forth. And so it's one way of making you a unique individual as to what type of glycolipids are found in your membranes. So let's take a look. Once again, come back to this image and focus in. There is the glycolipid. See, we've got our tails, the lipid part, and then the sugar sticking out here. And then some molecule will be able to recognize that and be able to respond to that, or not recognize it and not respond to it. And you'll talk more about that kind of thing when you do the chapter on the immune system. But those are marker proteins. Marker molecules that are found on the outside of the cell only. Okay. Alrighty. Okay. So, what else can we say? So that's the membrane part, but the membrane is composed of more than the lipids. So let's talk about membrane proteins. Membrane proteins. When we look at membrane proteins, there are two types. Number one, there are integral proteins. And number two, we have peripheral proteins. And we looked at those when we were looking at that last overhead of the cell membrane. Okay. So integral proteins, what they do is they're going to extend across the membrane. So they're found on either side of the membrane. So if this is my lipid layer, and I'm not going to go into the details with the little heads, but what we'd see is a protein that spans the whole length of the membrane. That's your integral protein. Okay. Some of these are what we call glycoproteins. And what does that mean? That means we have a sugar attached to a protein. And we'll talk about the significance of that a little bit in just a little while. Okay, so we can add sugars to those or not. They don't all have, the integral proteins don't have, all of them are not attached with sugars. Okay, so you have the integral proteins. Okay. Then we have the peripheral proteins. So the peripheral proteins do not cross the membrane. And they would be only found on one side. So they're only found on one side. Either the outer surface or the inner surface. Okay. All right, so the proteins give the plasma membrane functionality. So many of the things that we expect and find the cell membrane doing for the cell is the result of the proteins and the kinds of proteins that are embedded in that membrane. So with that in mind, let's go ahead and take a look at some examples, broad examples of the kinds of proteins that are found in the membranes. And to do that, I'm going to utilize, and I'll come back to this, we'll talk about this. I'm going to utilize this image over and over again. And so as we go through this, we'll talk about so-called ion channel proteins. And you can see that is an integral protein. It goes all the way across. We'll describe that. We'll talk about transporter proteins. We'll talk about receptor proteins. We'll look at enzymes and cell marker proteins and linker proteins. So those are the basic categories in which these proteins will fall under. And of course, within each category, we can have many variations. So there are many variations of ion channels, many variations of transporter molecules or proteins, okay? So let's go ahead and begin that process. So, and we'll title this part of the lecture as the roles of proteins. And we'll start off with the channel proteins. Now, channel proteins are all integral. And what they do is they move specific substances across the membrane. In terms of directionality, it could be in or out of the cell, both ways. Many, though not all, are ion channels. And here is a key concept, a key fact that you should always keep in mind. Ion channels are specific to what ion can pass through them. So this is they are specific to each type of ion. So if you have, for the sake of argument, you have two kinds of ion channels, and I'll indicate that there are two kinds by different colors here. So let me find a different color. All right. So we have two different types of ion channels. One is specific for sodium. And then let's say the other one is specific for chloride. So chloride cannot use this channel, and sodium cannot use that channel. Okay. That's what I'm talking about when I talk about specificity. This is very important because by having this arrangement, the cell can control what is happening, what is moving across that cell membrane. So if every ion channel is capable of moving any of the different kinds of ions, the cell would have no control over the movement of ions back and forth across the cell membrane. So this idea of specificity is very important when we look at channel proteins. Specificity is the hallmark of channel proteins. And keep in mind that most, though not all, channel proteins are in fact ion channels. All right? So that's ion channels. Number two, transporter. Proteins. These are sometimes referred to as carrier proteins. And they are also, all of them are integral proteins. Okay. And again, what they're going to do is they're going to transport materials across the membrane by changing shape. So they transport substances by changing shape. Okay. And they are also specific. So the same thing that we talked about with ion channels holds true with transporter proteins. Not everything can be transported across the membrane by any protein. So every protein, transporter protein, is specific to what it can transport and what it cannot transport. So let's go ahead and let's see if I can do justice with a picture of how this might work. So I'm going to draw really two pictures, and one shows the protein capturing something. So we're going to have this arrangement. Okay. And what we're going to do is we're going to capture something that is exactly that shape. Okay. So that this is what we're transporting, okay, and it fits into that. And so this is, let's just say this is extracellular, and this is intracellular. Now, once that binding takes place, then what you're going to see is that protein will change shape. And I've made this particularly difficult for myself. And that thing then crosses and is released on the other side. So it changes, literally changes shape to move the substance across the membrane. Okay. That's an example of a transporter protein. That's how it's different than channel proteins. Channel proteins are like tunnels, windows through the membrane. This is not a window. It's more it's more of a situation that where something is changing shape. In some department stores, to get into the department store, there's these revolving doors. You step in, and then you push the door, and you revolve around, and then you're inside the store. Well, that's an example of a transporter protein, as opposed to a store that might just have an open doorway. You just walk in without any kind of a change in the shape of that doorway. Okay. So there you are, that's transport proteins. The third type of protein are receptor proteins. These are also integral. And what these are going to do, they're going to bind to specific molecules. And these molecules are referred to as ligands. That's what they bind to, are ligands. And then what happens is they will alter the function of the cell. The cell physiology changes. So, for example, when a ligand binds to a protein, it might cause the protein to start manufacturing a new type of protein, or to stop manufacturing a protein that it was making. All kinds of things are possible. It could make the cell more permeable to a particular type of ion or molecule. So lots of possibilities. The point is that in order to get that change, you have to have a molecule called a ligand bind to one of these receptor proteins, and then that causes the change to take place within the cell. Okay. The fourth type are enzymes, and hopefully by now, since we've lectured on this, you know what we're talking about when we talk about enzymes. These can be both integral or peripheral proteins. So both are possible. And so what these are going to do are they're going to catalyze reactions. And a really good example of something that would be an enzyme that's attached to the cell membrane are in the small intestine. Attached to the cells that line the small intestine are enzymes, and these are called the brush border enzymes. And as you will learn when you do the chapter on digestion, these brush border enzymes are doing the last step in the physical breakdown of your food. So they're attached to the cells that will eventually absorb this material that you're eating. And in the lumen of the small intestine, there's been a lot of digestion going on. These large molecules are being broken down into progressively smaller and smaller bits and pieces. But that last step, the last possible step before you absorb that, whatever it is, a protein, for example, is going to take place right on the cell surface using these brush border enzymes. That is an example of an enzyme attached to the cell membrane, and that is an example of what we've been talking about in terms of functionality of the cell and how these proteins provide functionality to the cells. Okay, so enzymes. A fifth type are cell identity marker proteins. These are the glycoproteins. So your body uses specific combinations of sugars to identify itself within the body. So this has a variety of functions. The best example I can give you is that it's a way that your immune system can identify self from non-self. So hopefully your immune system would not recognize identity marker proteins that belong to you. Otherwise, it would mount an immune defense against you, and there are diseases where that does happen. But under normal circumstances, that does not. But a invader, a pathogen, a bacterium, or whatever happens to be gotten into your body will display the same thing, cell identity marker proteins, and your immune system will be able to recognize those and then see that as a foreign substance in your body and try to do something about it. One example of what we're talking about here in cell identity marker proteins are the so-called major histocompatibility complex. Which is quite a mouthful, so we're going to abbreviate that, and this is convention MHC, Major Histone Compatibility Complex. And this is, when you hear stories about people searching for donors for liver transplants and so, they're looking at these marker proteins and comparing them to your own. And so the closer that these proteins resemble your own proteins, the better the fit that foreign liver or whatever happens that you're getting transplanted will be. So that's just one example. Another example would be the ABO blood types. These are determined by different types of markers, cell identity markers on red blood cells. So it's just another example of that type of protein. The final type of protein is the linker protein. And these can be integral and peripheral. So either one. And you do recognize that you can't be both at the same time. Okay. So integral and peripheral. And these act as attachment points. On the cell membrane. So they can anchor. Think of them as anchors in the cell membrane that you can attach things to. So, for example, you can have cell-to-cell connections as a result of these. And we are going to talk about those when we get into the chapter. That's the very next chapter on tissues. We'll talk about cell-to-cell connections. And those would be examples of linker proteins. Okay. But they also they help the cell to maintain its shape. So many of the cells, in fact, all the cells in your body have a particular shape because they have a particular function. And that shape is maintained as a result of proteins that are attaching to that membrane and holding the membrane in a particular configuration. They can also provide movement. A whole host of different functions are attached to linker proteins that are part of the cell membrane. All right, well, let's quickly come back to the image and review what we just talked about. We started off talking about ion channels. And as you can see, it's simply a doorway through the protein and to move a substance, more often than not, ions from one side of the membrane to the other. We also had transporter proteins, and these are going to move things across the cell membrane by changing shape. Personally, I think I did a better job at drawing a change in shape than they did, so suck on that. All right, then there are receptor proteins, and these receptor proteins are things that the ligand attach to, and then they change the function of the cell. They alter the cell's function in some way. So antidiuretic hormone is an example, and changes the kidneys cells to changes their water permeability in the cell membrane of certain cells that line the tubule of the kidney. All right. So enzymes, we talked about those. So this might be an example of a brush border enzyme. So here we have, so let's say, two sugars attached to each other. This is in your digestive tract. And this was part of a much larger carbohydrate. We've broken that down in the lumen of the gut. And now we're back to we're here to the last step. And we're going to break that bond between the two sugars and produce two simple sugars, to monosaccharides, and this is what gets absorbed into the body. Notice, by the way, the functionality of having this happen right there on the cell surface. So these sugars, the products that you want to absorb, are being produced right on the cell surface, and they are therefore easily captured and brought into the cell rather than be lost out here in that very large space of the small intestine. Here is my cell identity marker proteins. They're using MHC proteins, the ones we talked about, as an example of what we talked. So it distinguishes your cells from anyone else's cells. And so one of the classes we talked about here was these major histocompatibility proteins. Not the only example, just one example. And then finally we had these linker proteins where we're connecting things together and holding them in place. And they're showing the ability to hold three proteins in the right order on the cell membrane by linking them together through these linker proteins. But there are other functions too, as we'll see in particular when we want to attach one cell to another cell. We'll look at the various types of ways we can do that. All of those will be these linker proteins that we talked about. Okay. All righty, Rudy. Well, let's move on and talk about membrane permeability. So this is going to be a very important function of the membrane. We talked about this in a number of different contexts. We talked about this in one of the labs, for example, when we did the lab on osmosis. And let's take a look at that again in a little more detail. And so we're going to list this as membrane permeability. Okay. Remember that the membrane is selectively permeable. We use this term quite often, and what we're going to see is how this comes to play. Selectively permeable, meaning that some things pass through and some things do not. That selectivity is the result of two things: the nature of the plasma membrane, the phospholipid bilayer, and the nature of the proteins that are embedded in that phospholipid bilayer. So selectively permeable comes from two factors. Number one, the lipid bilayer. And number two, the proteins. The type and quantity of the proteins. Okay. All right, in order to discuss this idea of membrane permeability, we have to get at some other concepts, and then we'll apply that to this broader concept of membrane permeability. So we're going to talk about gradients. We're going to talk about gradients across the plasma membrane. Okay. When we look at gradients, what we're going to notice is that often, if not all the time, there is a concentration gradient across the membrane. So let's begin there, and let's talk about a concentration gradient. Okay. The way we're going to define a concentration gradient is the following: a difference in concentration of chemicals or substances across the membrane. And let's go ahead and draw a very simple example of what we're talking about. So here's my membrane. And I'm going to go ahead and I'm going to take this. So I've got a lot of this on this side. Okay. And I could do this all day. It's very cathartic, especially in these times. And then over here, not so much. I could also do the following. Let me find a good, let's try this. That shows up not too well. Let's do something different. This is good. Pink. I can do this. A lot of those. And not too much over there. So I've drawn two different concentration gradients, and I've deliberately drew them on different sides of the membrane. So I have a high concentration and a low concentration. That is a concentration gradient. Over here, I have low and I have high. That is a concentration gradient. Difference in concentration of chemicals or substances across the membrane. Very simple, straightforward. Okay. Now, the type of things that are differently concentrated across the membrane can be divided into two very broad categories. So let's develop this idea a little bit more and talk about what kinds of things are going to create a concentration gradient. Okay, so we can have an electrical gradient And an electrical gradient is just what it sounds like, a difference in electrical charges across the membrane. All right, that difference is called the membrane potential. Membrane potential. Why do you use the word potential here? And the answer to that is that difference in charges gives you the potential to do work. Just as the concentration gradient that we talked about earlier gives you the potential to do work. So we've got two things going on here. We have the concentration gradient, which is a difference in the segregation of different substances across the membrane. And we have a separation of charges across the membrane. And once again, we can draw an example. So typically, what you see with most of the cells in your body, and there tends to be positive charges on the outside, and let me do this, and negative charges on the inside. That's a separation of charges. Okay, so this is extracellular, and this is intracellular. All right. And what we do is we combine these two concepts. We combine the concept of a concentration gradient with the concept of an electrical gradient. And what we come up with is a electrochemical gradient. So if we combine the concentration gradient with the electrical gradient, concentration gradient, I forgot that. With the electrical gradient, we come up with the electrochemical gradient. Okay. Let's take a look at some examples now. We're going to use the textbook and just look at both these examples again. So here we go. So here is a concentration gradient. We've got a number of different chemicals that are segregated across the membrane, some more on one side and some less on the other side. We can see here potassium and carbon dioxide, and then oxygen and sodium, and you can see that there's this segregation across that membrane. Okay, and then we get this separation of charges across the membrane, with the inside of the cell typically being negative and the outside being positive. And by the way, you can literally measure that difference in charges, and they have little meters that they have designed and built, and you can get a little probe and you can probe it to the inside, and then the other probe is on the outside. You can measure the difference in electrical current or electrical potential across that membrane. So somewhere in the range of about minus 90 millivolts is the typical difference across that membrane. All right. So what's the point? Why are we talking about this, and what does this have to do with the membrane? Well, a couple of facts, if we write them down, will help us understand. So, flow down a concentration gradient. So, the movement of something from high concentration to low concentration can be taken advantage of if we create or have that concentration in the first place. So one of the benefits that we get out of having that concentration gradient is the fact that that the molecules that are segregated across the membrane want to move down their concentration gradient from high concentration to low concentration. And that gives us a number of potential things we can do with that. For example, we could do work. As they move down the concentration gradient, we can get them to move other molecules with them. And we'll talk more about that as we get into the types of proteins that are using this flow of from high concentration to low concentration. The other thing is that negative charges are attracted to positive charges. And again, that gives us certain advantages, because if we create this difference in charges across the membrane, the natural tendency built into that system is for the negative charges to be attracted to the positive charges. And they can move, if we allow them to move, through specialized types of proteins. And in so moving, we can get work done. Okay? What you'll see, for example, is that action potentials take advantage of this property right here. That is the things that allow you to think, to move your muscles, to secrete substances from your glands, for your nervous system, control your body. It's all done through action potentials. And those action potentials are only possible because we have created that difference in concentration across the membrane. So just to emphasize this a little bit, let me come back to that drawing and show you what I'm talking about. So in this image, I have, let's say, oxygen. I have a lot of oxygen here, and on the other hand, I have a lot of carbon dioxide here. So this oxygen wants to go from high concentration to low concentration, and that would be into the cell where we need it. And carbon dioxide, which is building up inside the cell, will want to go in the opposite direction. We'll want to move out of the cell and into the interstitial fluid. So just by the nature of that concentration gradient, we're taking advantage of that, moving oxygen into our cells where we need them, and carbon dioxide, which is a waste product, out of our cells through that very simple idea of concentration gradient. The same can happen here. So I have these negative charges, positive charges. They're attracted to each other. If I produce the right protein in here, I can have those positive charges move across the membrane. And when they do that, I can produce certain types of functionality out of that. And I won't go into the details there because that really is a whole chapter that you're going to cover in 40B. So that's just a couple of examples of how this all works out. Okay. All right, so that gives us a little bit of background. And so what I'm going to do now with that background is I'm going to talk about membrane physiology. And what we see is that by the very nature of the membrane, we get three very important physiological properties. So we get three physiological properties out of what we have been describing so far. That is to say, out of how the membrane is put together, including its proteins, and these electrochemical gradient. I'm going to quickly list them and then we'll go into more detail. So one of these properties is, number one, communication. This is a critical idea. Your body cells need to communicate with one another in order to coordinate the activities which maintain homeostasis inside the body. So communication is a very important property, and it comes out of, as I've already said, the properties what we have just described. We also can produce this electrochemical gradient. Okay. And we can also, by the nature of that membrane transport, we can be selective in our transport of substances, okay, across the membrane. Okay. Oakley dokleys. Let's talk about the first two, or actually let's talk about the first one very quickly. And we'll talk a little bit about the electrochemical gradient as well. And then we'll also talk about transport across the membrane in various processes that we do that. So let's begin with communication. And we won't spend a lot of time here, but we do need to address it in somewhat more detail than we have already. So communication. Okay. So cellular communication, as I mentioned, very important. If you're going to coordinate body activities, and let's face it, basically what you're doing is coordinating the activity of all those trillions of cells we talked about, you have to have some mechanism to do that. Now, cellular communication is going to be between cells, okay, between your cells and foreign cells, that is bacteria. Okay. And it's going to involve often a ligand of some sort is going to be involved. So ligands are more involved in this process here between cells. We don't need ligands to for your immune system to identify and communicate and attack foreign cells in your body. Okay, and so we talked about this when we looked at the different types of proteins, and you should go back probably and review the different types of proteins and try to figure out which one of those fit into this category. It's not that difficult. All right, so the next one is the electrochemical gradient. And this is as a result of the difference in the concentration of charges and substances across the membrane. Okay. Yes, quite often they are very closely connected because the things that are carrying the charges are in fact substances such as ions. Okay. So let's take a look at a chart, and this chart is actually available in your textbook under the chapter Electrolytes in Body Fluids, which is chapter 27. So I just went ahead because I knew it was there, and I pulled this out so we could look at it to give us an example of this electrochemical gradient. So let's do this. And let's zoom in a little bit So first of all, let's interpret what we're looking at. So over here, just focus on the red bar is your blood plasma. So we're going to look at a variety of different things and their concentration in 3 different areas: blood plasma, interstitial fluid, and intracellular fluid. And what we want to do is we want to just point out differences in the concentration within these various fluid compartments. And keeping in mind, in intracellular, we're talking about inside the cell. Interstitial, of course, is the fluid that surrounds the cell, and blood plasma is the fluid that's found in the circulatory system. So let's take a look at sodium over here real quick. Here's sodium. So again, the brown bar or tan bar represents the cell. So sodium has a very low concentration inside the cell, but very high concentration outside the cell. High outside, low inside. And of course, sodium also has a charge. So that means that we have a separation of charges here. Potassium is just the opposite. High concentration on the inside of the cell and very low concentration on the outside of the cell. Here's another one. Magnesium, very high concentration on the inside of the cell, low concentration on the outside of the cell. Chloride, low concentration on the inside, high concentration on the outside. This is bicarbonate. These are, again, there's a difference, but not as dramatic as we've seen in the past. Phosphate groups. So a tremendous amount inside the cells and very little outside the cell. Same as with the sulfide groups and the proteins. So as I pointed out to you, when you look inside the cell, the inside of the cell tends to be negative and the outside tends to be positive. Okay? So where is that difference in charge coming from? So this, and this tends to make the cell negative. Okay? And this tends to make the cell positive. That's our separation of electrical charges. So this difference this difference over here, this difference of sodium as compared to inside the cell. So there's much more sodium on the outside of the cell than on the inside of the cell. And the reverse is true over here. These all are negatively charged. Proteins tend to have a negative charge. These 3 combinations tend to make the inside of the cell more negative, and this tends to add to the positive charge on the outside of the cell. Okay, so there's our electrochemical gradient. All right. So let's get a little bit of that information down into our notes. So intracellular. As I mentioned, proteins. The organic phosphates. And sulfates have a higher concentration. Making inside the cell more negative compared to the outside. Okay. Extracellular is the sodium, higher concentration. Making the extracellular more positive than the intracellular. Fluid. Okay. All right. So, and that difference then is the membrane potential. You're separating positive and negative charges, and they are attracted to each other. So if they want to get back together, then you can make them do work, or you can produce some sort of physiological process, such as an action potential. Oakley dokley's. Let's move on, and let's talk about transport across the membrane. So we're going to do transport across the membrane. Transport across the membrane. And I'm thinking, just based on my fatigue level here, that this might be a good time to break this lecture off, and I will produce a second lecture which will cover the rest of this material. Yes, I see that I bit off more than I wanted to chew here in the first lecture. So let's take a break right here, and next time you see me, I will be talking about transport across the cell membrane. So have a good evening, and I will certainly see you in the near future. And can I do this? I think I can do this. Yay.